All right, folks, welcome to part 2 of lecture 4 of GVZ. Now we're going to move into reconstructing phylogeny. So we've looked at a lot of phylogenetic trees. We've discussed their structure and some properties of them and the terminology associated with them. We've discussed how they're applied in terms of our higher level taxonomies with phylogenetic nomenclature. Now we're going to talk about how we actually get the trees. So the kind of analysis you do of data to produce those ball and stick diagrams that are so important for evolution and for understanding pretty much everything about biology. So the first thing I want to talk about is just the data, the raw materials that are used to reconstruct relationships. We generally think of these in terms of characters, which is a certain element that can vary, and then character states, which are the different instantiations of that character. So, for example, really straightforward example is a DNA base, a DNA site, where your different options are, of course, adenine, guanine, cytosine, and thymine. So those are your 4 different character states for that one character. But really, anything that has a set of properties that I'm going to talk about in the next slide can work. In addition to genes, there's all kinds of other genetic data like gene order. In your mitochondria, among different species, genes will flip places, and that's a good marker. Morphological traits like having presence or absence of a bone. Kind of macromolecular traits like chromosome number, chromosome structure. And then, behavioral traits, things like parental care, what kind of nest you build in terms of, using dirt or using vegetation. These can all be good markers. And it's a really important take home that, one, that, all of this kinds of data is acceptable and useful for reconstructing phylogeny. And two, something that I don't have up on this slide, but there's never been shown that one particular kind of data is more reliable than another. So like most people, their first pass is to think, okay, certainly the DNA are going to work best. Turns out that's not the case. They don't really work any better than morphological data. DNA sequence data are, our most common source of data that we collect these days because of the huge numbers of characters you can get. The largest morphological data sets, if you're talking about looking at, say, bones and external anatomy, are on the scale of, a couple hundred, 300 characters. Whereas, of course, DNA sequence data sets, you can get hundreds of thousands of characters. And so that's really the advantage of molecular data. It's not reliability, right? It's just having this sheer number of data that can help you. So what are you looking for? You want characters that are heritable, character states that are heritable. So that's passed on to offspring. And then you want characters that are independent from each other. And by this I mean that a change in one of the characters doesn't affect the change in the other. It's an underlying assumption of most methods that we use that each character is an independent item of information. Now that assumption gets violated to differing degrees all the time. But we want to get as independent as we can possibly get. So, for example, if you were to, so that sample of limbs on the right is showing, homology between one of the long bones and the distal limb in red there. But, think of if you included both that femur or forelimb and then the red long bone, had each of those be a marker for phylogeny, and you'd had, say the length of one and the length of the other. Those things are going to be correlated. And when they're correlated, that means they're violating that independence assumption, so you wouldn't use both of those traits. Variable at an appropriate level. And so what I mean by this is that you need traits that don't change too rapidly, evolutionarily, in the group of interest, but also don't change too slowly. And this is completely connected to what clade you're evaluating and, kind of how recent the evolutionary splits are. But just to give an example, like, presence or absence of fur is not a useful trait for figuring out the relationships within mammals, right? It supports the clade, But with mammals, they all have fur, so that's not very helpful. Whereas something like, variation at the cytochrome B gene would be helpful. And then the last thing I'll mention is that when you're enumerating character states within a character, you're essentially making a hypothesis of homology. And so, we can say that distal bone there is homologous between those three things, but that's a hypothesis we're making, right? And it's the same thing with DNA. The issue there is alignment and making sure you have your sites lined up in a way that captures the historical signal. All right, so phylogenetic analyses generally have two components, and this was actually something that was confused for a long time in phylogenetic biology, but is now really well, since the late 90s, I would say, figured out. One of the aspects of a phylogenetic analysis is the optimality criterion. This is a quantitative, it's a score that indicates which is the best set of relationships. So if you're considering two trees and one has a score of 16 and one has a score of 17, and lower is better, then the one with 16 is preferred. And you apply whatever your optimality rule is across all of the trees that you're evaluating. The second aspect is tree search. And so this is once you decide on a optimality criterion, and I'm going to review those next, you still have to evaluate trees. And this is something that it's not immediately obvious why this is an issue. But as I'll show you in a moment, the number of solutions to the phylogeny problem, once you have not very many taxa, not very many branches, it becomes too hard to figure out computationally. And that's not to say we can't enumerate the exact number of solutions. We can. We just can't find which of those is the best. That's a really difficult combinatorial problem. But that's stuff I'll get to in just a minute after I tell you a bit more about optimality criteria. So we will discuss three different optimality criteria. Remember, these are quantitative ratings for which tree is best. A venerable and very simple method that I'm almost certainly going to show you kind of how to do it yourself, you can do it by hand, is the parsimony method. And this, if you're familiar with the word, then it's a very intuitive method where you're minimizing the number of evolutionary changes. So you're choosing the tree, you're preferring the tree that has the fewest number of changes on it evolutionarily. So looking at this example here, chimps, humans, gorillas, and lemurs, if you put chimp and human together, then that tree only requires one change to happen. So for example, from a C to an A. If you put gorilla and human together, then you're requiring at least two changes. And if you see that immediately, that's great, you're ahead of the game. If not, we're going to go over this kind of thing more in just a minute. Another criterion is distance. And this involves finding, just like it sounds, the evolutionary difference between two species or two individuals. So you take a data set here shown for this data set, human, chimp, gorilla, and lemur. The columns are the characters, the DNA sites. And then if you are looking up and down vertically in a column, you're looking at the different DNA bases for each of those species. So for the first character, human and chimp have an A, gorilla has a G, and lemur has a C. And so what we do is come up with some measure of the relative similarity of those things. And a really crude one would be just to count up differences. So say, if we look at, say, human, look at, say, human and chimp. And at the first site, they both have an A. The second, they both have a C. The third, they both have a C. And the fourth, they differ, C to G. So we could say, okay, scale difference between them is 1 out of 4 or 25%, and then you use that information to put those two species together on a tree. And so you can look at, if you compare the tree on the right to the pairwise distance matrix in the lower left and the data set above, you should be able to track that, the distance from lemur to those other taxa is greater than any of the other between taxa differences, because it's differing in all those different sites. The kinds of methods that are most used today are explicitly statistical methods. This is important because it allows them to use a very particular and specified model of the evolutionary process it's incorporating. So the two approaches that are statistical are likelihood approaches and Bayesian approaches. These correspond to philosophically very different schools of statistics. In terms of their application, they tend to get the same results because their basis is really the likelihood function. And the way this works is if you look at those four kind of equations there, look at the one in the upper left, and it's showing a probability of this distribution AACC across that phylogenetic tree, comma, and then HKY as a type of evolutionary model, and then the number over to the right, then the 0.0000021 is that probability. And so this is, the important things are that it's explicitly statistical, unlike parsimony and distance, which you can present them in statistical ways, but they're not operating from the get-go with that. And then second, that it's easy to incorporate really specific models of the evolutionary process. Okay, so we talked about optimality criteria, parsimony, distance, and the statistical approaches, likelihood and Bayesianism. Let's talk about tree search now and communicate why this is such a difficult problem. So let's say you have three taxa that you want to figure out the relationships of. For example, like what we've been using, the chimps, humans, and gorillas. If you have three taxa like that, three species, how many different sets of ways can you show those relationships? How many different relationships are there for those three taxa if you have that rooted tree and the only thing you're worried about is the relative dichotomous splits? For three taxa, the number of potential evolutionary relationships is 3. You can have chimps and humans sharing a evolutionary history separate from gorilla. You can have chimps and gorillas sharing an evolutionary history separate from humans, or you can have gorillas and humans sharing an evolutionary history. It shouldn't be separate from gorillas there. It should be separate from chimps. Sorry about that. All right, so what about if we have 4 taxa? So we just learned that for 3 taxa, we have 3 different possibilities. What if we have 4 taxa? 4 taxa, now all of a sudden we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 options. Okay. So we went up and we increased our taxa by 1, but we increased our possible solutions by 12, right? That keeps going. For 3 taxa, the number of trees is 3. For 4, it's 15. For 5, it's 105. 6 is 945. This is an exponential increase, right? The numbers are increasing linearly. You're just adding 1. And the number of trees is increasing by a multiplier each time. This becomes intractable really quickly. A problem with 100 species, which, think about any of your favorite groups, birds with 10,000 species. 100 species, and you have a problem that has more solutions than the number of atoms in the known universe. And I know that sounds like some sort of way of saying it's a really, really big number, but that number is much larger than any of the hypothesized number of atoms in the universe that have been suggested. And that means that there are too many solutions for even the best supercomputers to evaluate. Again, we can enumerate that exact number. If you want to ask how many trees there are for 1 billion taxa, you can calculate that. The issue is figuring out what the best of those is. So applying your optimality criterion to all those different trees is what becomes impossible. Okay, so this is a marking point where we're going to move on to our next thing. And this is to do an actual example parsimony analysis. So we'll do a super simple treatment here where the idea is for you to just see how this works, but also be able to do it yourself in terms of finding the best tree and also what's called the tree length, which is the optimality criterion for parsimony. So these are the steps for a parsimony analysis. First, you have some set of candidate trees you're evaluating. Our problem is going to have 3 taxa that we're interested in, and so we're going to have 3 potential solutions, as we just saw. On each of those trees, you find the minimum number of changes for each character on each of those trees. Then you add them up for each tree, and then you prefer the tree that has the fewest changes. Now, probably what I just described to you is confusing, and that's okay because we're going to go through these steps. All right, so the first thing we need in our example parsimony analysis is some actual data. Here is a set of 3 characters. One is endothermy versus ectothermy, and we're coding these using 0s and 1s, as is common in the field. Our next character is, do you have gills or do you have lungs? And our third character does the species we're evaluating, does it have paired pectoral fins or does it not have them? All right, so here's our data matrix of 3 species we're interested in evaluating, and then an additional species we're using to root the tree. So we're interested in the relationships of humans, coelacanths, and dolphins. We call that the in-group. Any phylogenetic analysis actually requires some assumption about relationships, curiously enough. You have to make an assumption about something that's outside of your relationships of interest. So we're going to assume that the lamprey is not nested within the relationships of humans, coelacanths, and dolphins. It's going to act as what's called our outgroup. And the outgroup is included to root the tree. So you can see your characters there in the upper right, your data matrix on the left, and then in the lower right, there's our options. So let's map this out and do our steps for a parsimony analysis. First step, find the minimum number of changes for each character on each candidate tree. So let's start with character 1, and let's map out the traits at the tip of the tree. So we have human, dolphin, coelacanth, and lamprey. That first tree has humans and dolphins together, coelacanth outside of them, and then lamprey is always going to be the sister group, the outside group to all those others. We map out the traits where human and dolphin have a 0, and coelacanth and lamprey have a 1. If we assume that the root state, the state at the bottom of the tree, the state at the oldest part of the tree is the same state as the lamprey, then you can arrange the evolutionary changes on this tree such that only one of those changes occurs. You can say that in the shared ancestor of humans and dolphins, you have a change from 1 to 0 that's passed on. If you look at the second tree and make the same assumptions, that is that there's a 1 down at the base, all the taxa at the tips have those states, there's no way to arrange changes on this tree such that fewer than 2 changes occur. And just as a sample of that, I'm showing a change from 1 to 0 going out to human, and a change from 1 to 0 going out to the dolphin. And what you should do, if once you get comfortable with this, is try some other arrangements and see if you can find fewer changes. You should be able to find more changes. You can arrange the evolution such that you're getting more than 2, but not fewer. And we see the same distribution on the tree on the right. However you slice it, you have a minimum of 2 changes. For character 2, we can do the same sort of mapping, where in this case, character 2, our lamprey, the outgroup, has a 0. We assume that's the state down at the root. We can arrange the changes on the tree for the tree on the left to just having 1 change on the branch going to human and dolphin. If we look at the other 2 trees, then there's no way to arrange the traits such that fewer than 2 occur. The third trait, or the third data point, the third character, in this case has coelacanths and dolphins with the same state. And so coelacanths and dolphins with a close relationship is shown in that middle tree. And so if we take that middle tree and assume that there's a 1 in the outgroup, we can say there is a 1 to 0 change on the branch going to dolphin and coelacanth. The other trees require at least 2 changes. All right? Now, we'll take the sum across all of those changes. So look at that first tree. Character 1 has a length of 1, or a number of steps of 1. Character 2 has a length of 1. Character 3 has a length of 2. That adds up to 4. So that has 4. The next tree has 5. The tree on the right is the worst solution. It has 6. Because the next final step is to prefer the tree with the fewest changes, the lowest number, and that's that first tree there. So the tree with the length of 4 is the tree that we prefer. Now, this is often one of the more difficult things in this first part of the course, although I feel like I keep saying that, but it's one that a lot of people have trouble with. I'll give you some practice on it, some solved problems that you can try and do, and there's some practice in your reading as well. Okay, so our last big section is going to be on reconstructing ancestral states. So this involves taking some group that you're interested in. In this case, this set of amniote taxa are reptiles and are mammals. And you want to know what the root state was, what the ancestral state was at some particular part of the tree. So let's take this tree and let's map out the outer covering of the exterior of these different taxa. Birds have feathers, crocodilians, turtles, and lizards have scales, mammals have fur, and amphibians have skin. And so we're interested in, what is the ancestral state for reptiles? So the group that includes lizards, turtles, crocs, and birds, that branch leading to that group, what condition did that have? Did it have feathers? Did it have scales? Did it have fur? Did it have skin? In some cases, we have fossil evidence that can help us with this a lot. But in other cases, we're just left with information on extant taxa. This particular version of this problem is a fairly difficult one. So let me show you 2 rules. One of them that should make sense and be intuitive, and then the other just kind of being the flip side of that. So if all members of a clade have some state, here I'm calling it clade X and state A, then state A is assumed to be the ancestral state for clade X. So let's look at, for example, the amniotic egg. All of those taxa shown there, birds, crocs, turtles, lizards, and then, think of mammals as laying eggs, but the ancestors of mammals actually did have shelled eggs. The outgroup has aquatic eggs. Following our rule, it's pretty straightforward to infer that the ancestor of reptiles had an amniotic egg. All the members of our reptile clade have an amniotic egg, and so we're going to infer that the branch leading to reptiles had an amniotic egg. Straightforward. But our initial example here for reconstructing ancestral states is not so straightforward. And that's where we have our second rule for the purposes of this course. If some members of clade X have state A, if only some of them have it, then it's complicated. Then it becomes a fairly difficult and uncertain quantitative statistical problem in a lot of cases. So for our purposes here, if everything in the clade has some particular state, we can assume the ancestral state is the same. But if you have some mix of states, if you're dealing with a lizard clade where some things are viviparous and some things are oviparous, you're not going to be able, for our purposes, you're not going to be able to infer the root state because it would take some serious computations and some extra knowledge that we don't necessarily have with things like branch lengths, and as I said, like fossil information would help as well. So first case, everything has it. Easy. Let's use that in this course. second case, there's some variation, then it becomes very difficult to infer ancestral states. Okay, entering the home stretch here for our first recorded lecture. I think I will go quickly through this ecology terminology and just talk to you about that a bit, and then tell you about our reading, and then we will be good. Okay, so as with other times when I'm just kind of throwing up terms, these are words you need to know, a lot of which I'm sure you already know, and probably already know well enough for the purposes of this course. Probably some of these, though, you don't quite know in a way that's going to be helpful. These are words we're going to be using here, and I just want to, same thing as looking at, proximal and distal, I just want to make sure we're all on the same page so that when you see the word assemblage on a test, you'll know what we mean. So, an ecosystem. An ecosystem is just the sum total of the interacting organismal and abiotic parts. So think of like an island, and you have island with certain geographic structures on it, rocks, sand, trees, and that. And you also have a series of living things on it. That whole thing is the ecosystem. Sometimes ecosystems are considered, very large, like, the Sahara Desert or something. The kind of edges of ecosystems can be fuzzy, both conceptually and operationally. But we won't worry about that. We won't have to worry about that in here. A community, for our purposes, is going to be all of the living species and living individuals in a particular area, often an ecosystem. An assemblage is a set of, it's like a community on a smaller scale of close relatives. So, for example, we look at our picture here. If we were to analyze all the turtles in this pond, so not the turtles and the fish and the insects and everything, but just the turtles, we might say we're analyzing an assemblage. Whereas if we're looking at all those things together, we'd probably talk about it more like a community. Is this something of a continuum? I would argue yes. Other people might argue differently. Whatever minutiae there are in those terms, you don't have to worry about. Habitat and microhabitat, you should have some sense of, habitat just being the areas that an organism lives in. This thing lives arboreally, up in trees. Those are some other terms that maybe you'll make sure that you know, like arboreal and saxicolous and things like that. The microhabitat, as you might guess, refers to just the very particular part of the habitat that you're living in. So if the habitat can, if we think of habitat even a little bit more broadly than I just mentioned, so think of a habitat being, say, the area within this picture here. Your microhabitat might be one of those trees, for example. Again, you can probably tell by my treatment of these terms that it's not something where we're going to be worried about very precise differences here. Another term that we're going to use that I think could sometimes be tricky is the idea of a niche. The niche has multiple definitions in formal ecology, at least 3. For our purposes, we're just going to think of a niche as sort of what role an individual or a species fulfills in its environment. And, role sounds kind of anthropomorphic, but just think of like what it's doing there. Something might have a niche where it lives in trees and eats insects and is active nocturnally, something like that. The fundamental versus the realized niche. The fundamental niche is the areas that an organism would occupy if it didn't have any predation affecting it or any competition keeping it out of certain areas. There's always predation and competition, so your realized niche is what you're actually doing. And that leads to the ideas of niche overlap and niche partitioning. So niche overlap, just as you'd expect, refers to things, inhabiting the same ecological scope. So, things that are in the same pond, for example, and acting near the bottom of that pond. Partitioning is the idea in ecology that things tend to work to maximize their resources and to minimize their competition. So when you look at a community of frogs, and some of them are always in the water, and some of them are on the bank, and some of them are in the trees, they're partitioning that area spatially. When you're looking at a series of frogs that are active, some are active early in the summer, some in the middle, some late, they're partitioning by time. This example here is from a paper in, I think, 2022. I can't quite read it with my bad eyesight, but it's showing a really cool case of partitioning prey where carcasses, so dead animals that a lot of things eat, the time when things are eating them gets split up. And what these authors found was that the carrion eaters among birds tended to operate during the day, and the carrion eaters among mammals tend to operate at night. And this allowed them to avoid each other in terms of competition for the carcass, but then also in terms of any extra predation that might be happening. So they're partitioning that food niche and doing it in a temporal way. Okay, so that is it with us getting our feet wet for the material for our first recorded lecture. Let me quickly go over your reading. So again, as I've mentioned, that phylogenetic nomenclature is difficult, and people have trouble with it. And there's people who, miss that question 3 tests in a row. So what I want you to do to get some additional insight on this is to go to the actual phylocode, and I'll give you a link for that. So this is the document, the manifesto of the people who advocate for this sort of thing. I want you to read the preface, the preamble, their Division 1, their Division 2, Chapter 1, and then also Division 2, Chapter 4, Article 9. This isn't a lot of pages, it isn't a lot of words, and it will help you. So read that as reinforcement. This paper by Tim Rowe is one that I referenced when I was talking about taxonomy. And I really want you to get a handle on that definition versus diagnosis distinction. Again, very similar to our earlier conceptual operational distinction when we talked about species. So this is a paper by Tim Rowe. There's a lot in there. Some of it's kind of dense. Please read the introduction and then read his section on definitions and diagnoses, and you should be fine. This is a website link I'm going to give you about reconstructing evolutionary relationships. It's out of Berkeley, so it's a reputable place. And so I'll give you a link to that where you can go and practice and get a better feel for how to reconstruct the relationships, evolutionary relationships or phylogeny of things based on data that you have. All right, so that will be it for Lecture 4. I will be in touch, and I'll see you soon.